Hello everyone, welcome to the Civil Service Examination 2020 Smart Crash Course, and this is the fifth lecture in the series related with science and technology, and today in this lecture we are going to cover biotechnology. Let's go ahead. So the first and the foremost thing that we are going to discuss is related with the basics of biotechnology, and in the basics of biotechnology, first and foremost thing that we are going to discuss is the basis of genetic material, that is DNA or deoxyribonucleic acid. See, the DNA is the genetic material of the maximum number of organisms. So, a very important point here that you have to remember is we are not considering all organisms but maximum number of organisms. However, if we say organisms, yes, DNA is the genetic material for all organisms but not all substances. For example, viruses. Now, viruses are some of the like pseudo organisms which are having even RNA as their genetic material. Like, for example, you have heard about the HIV virus, you have heard about the COVID-19 virus. Alright, so the novel coronavirus, which is recently in news, that is also not having the DNA as its genetic material, rather, it is the RNA. So, in total, we can say that the nucleic acid forms the genetic material of all substances on the earth, and it is particularly the DNA which is for the living organisms, and mostly in RNA, in viruses, you can find RNA as well as DNA as the genetic material. Alright, so nutshell over here that we have to discuss is that the DNA is the genetic material. In organisms, like particularly the eukaryotic organisms or the developed organisms, the DNA, they form something what is known as chromosome. Alright, so chromosome is the synthesis of the DNA in the form of a clustered material that is basically known as chromosome. और ये जो chromosome होता है, this chromosome determine the characteristic of the individual. So all the physical characteristic, mental characteristics, they are derived from the chromosomes that are contained in an organism. For example, if I have to give you an example here, the chromosome number 1 in the human, that is related with your susceptibility to malaria. Like, for example, it is your chromosome number 1 that is going to determine if you will be suffering from malaria in near future or not, or some other kind of diseases are also present on different kind of chromosomes. Similarly, your chromosome number 16, it deals with what hair color you will have. Alright, so your chromosome number 16 will have genetic material from your mother's side, your father's side. If suppose your mother has blonde hair and your father has brown hair, so there's a 50% probability that you will be having the blonde hair or you will be having the brown hair, but that will be entirely determined by the chromosome number 16. So it is the DNA that is taking the form of chromosomes that give you the physical characteristics. Alright, let's proceed ahead. Okay, so in human body there are two copies of 23 chromosomes. That means that there are total 46 chromosomes, and 23 chromosomes are coming from the mother side. That is, one entire copy of 23 chromosome is coming from the mother side. All right, and there is one copy of chromosome, that is 23 chromosomes, coming from the father side. All right, so that is how your normal somatic cells, they have some 46 chromosomes in total. All right, and these chromosomes, they are present in every cell of the human body. That is, there are 46 chromosomes in every cell of the human body. There are certain conditions under which the individual may not have 46 chromosomes, but then there is chromosome related disease which we are not covering over here. All right, but I'm sure that you must have covered this thing in your static class. So next thing we have to discuss is that there are 46 chromosomes in human cell, and these 46 chromosomes have 6 billion base pairs of DNA. That is 3 billion base pairs coming from your father side, 3 billion base pairs coming from your mother side, and in total there are 6 billion base pairs present in a human cell. Alright, now what are base pairs? To understand the base pairs, we'll have to first understand what is the structure of the DNA. So before I actually go and describe the structure, you have to understand that what a DNA comprises of. So DNA stands for deoxyribonucleic acid. So there is a nucleotide that is the fundamental particle of a DNA. Alright, so it is a nucleotide that is the fundamental particle of a DNA, and this nucleotide consists of a sugar backbone, attached with it a nitrogenous base. Alright, so this is a nucleotide. So DNA consists of 4 nitrogenous bases and attached to a sugar phosphate strand. That is, one nitrogenous base associated with a sugar phosphate will make one nucleotide. So it is a kind of polymer having multiple nucleotide attached with each other, and that is how a DNA looks like. So let's go and further discuss the structure of the DNA. As I told you, that the nucleotide, which is the founding particle of a DNA, it consists of four nitrogenous bases. The four nitrogenous bases consist of adenine, that has representation through A, thymine, that has representation through C, guanine, that has representation through G, and cytosine, that has representation through C. It is to be understood very clearly that ATGC, they are known as the nitrogenous bases. They are the nitrogenous bases which attach themselves into a strand of made up of sugar and phosphate. Now, it is a very interesting property of the DNA that the adenine and guanine, the two nitrogenous bases, they have the tendency that they form hydrogen bonds with thymine and cytosine respectively. That is, adenine will associate with thymine, and guanine will associate with cytosine, and vice versa. That is, thymine has the propensity to form hydrogen bond with adenine, and cytosine has the propensity to form hydrogen bond with guanine. All right. So therefore, in a way, the DNA will consist of two sugar strands parallel to each other, on which nitrogenous bases will be arranged in such a manner that the opposite sugar strand will have corresponding complementary nitrogenous base. In front of adenine there will be thymine, in front of thymine there will be adenine, in front of guanine there will be cytosine, in front of cytosine there will be guanine. So this way there are two parallel sugar strands on which you will find the nitrogenous bases in a complementary manner, such as one nitrogenous base adenine will be complemented by thymine, cytosine will be complemented by guanine, and then these two sugar strands which are parallel, they are twisted in such a manner that they give DNA a structure which is particularly known as the double helical structure, which was discovered by the famous biotechnologist Watson and Crick, and therefore Watson and Crick were given the Nobel Prize for discovering this double helical structure of DNA. So I think that the structure of DNA is quite clear to you. Let's proceed further. Alright, so the next thing we have to discuss is DNA profiling or DNA fingerprinting. So DNA, you know that DNA is something which is peculiar to almost every human being, and it is unique to every human being. So what is actually happening? The almost 99.9% of all human DNA is same, but 0.1% of everyone human DNA varies. That means that every individual is going to have a unique arrangement of the base pairs in the DNA, and therefore, since it is unique, therefore this uniqueness can be used as a biometric parameter for biometric database preparation, such that everybody's DNA, if it can be profiled, if it can be fingerprinted, if it can be associated with every individual will have his or her own DNA profiling or DNA fingerprinting. Alright, so this process of creation of a DNA database or biometric DNA database is basically known as DNA profiling or DNA fingerprinting. It is very important to identify, it is very important in various circumstances. For example, in crime scene. In crime scene, you can very easily retrieve the DNA of the person who has committed the crime, and from the suspects you can match the DNA, and then you can conclude that who is the culprit behind the crime. Similarly, DNA fingerprinting or DNA profiling is also of very utter importance in cases related with lineage or parentage. Alright, so this is how DNA profiling and DNA fingerprinting are founding a greater place in today's biotechnology. The next thing we have to discuss is genome. So genome is nothing but it is the full set of DNA present in an individual organism, and this is known as genome. That is, entire genetic material, if it is written down in a code, that entire genetic material is referred to as genome. And when you write down that code, when you determine the linear order of the arrangement of the base pairs on that genetic material, this process is known by the name genome sequencing. All right. So, what is genome? Genome is nothing but it is the total genetic material present in a human being. And then, so, basically, what you are doing is you are profiling, you are decoding the entire 46 human chromosome, entire genetic material, and arrangement of the base pairs on that genetic material. This process of identifying the base pair arrangement of genome is known as genome sequencing, and genome is nothing but it is the full set of DNA present in an individual organism. Now we are proceeding further and we'll discuss something that is known as mitochondrial DNA. So basically in human body, the DNA is found in two places. One is the nucleus and second is mitochondria. All right. So if you look at the process of fertilization in this diagram here, this is the egg of the mother. This is the nucleus of the egg of the mother that contains 23 chromosomes from the mother side, and then there is a mitochondria. As I already told you that a DNA or one chromosome is present in mitochondria, 23 chromosomes are present in nucleus of the egg. This one chromosome of mitochondria has nothing to do with the genetic characteristics of the child. It is only responsible for processing of certain proteins for the process of metabolism. All right, it is nothing to do, this mitochondrial DNA, it do not participate in genetic mechanism. Remember this thing, very important. Nucleus DNA is for genetic purpose, that is the characteristic feature of the child, but this mitochondrial DNA is not related with genetic purpose, it is only related with metabolism. All right. So when this sperm, which also contains 23 chromosomes, it will come and fertilize the egg. The individual will grow, and this individual will be having 23 chromosomes from the mother side and 23 chromosomes from the father side. Therefore, 46 chromosomes in total. All right. So this is basically the genetic. This is basically the genetic information that is present in the cell. One in the nucleus and then in the mitochondria. The mitochondria is having one chromosome, the nucleus is having 23 chromosomes. 23 chromosomes of nucleus are associated with determining the genetic characteristics, whereas in terms of the mitochondria, the mitochondrial one chromosome is not participating in genetic mechanism. Now what happened that many a time the chromosome present in mitochondria, it may consist of certain genes that can cause cancer. For example, yeah, any kind of disease. So mitochondrial DNA is quite probable may result into some kind of diseases. So what can be done is this mitochondrial DNA, which may result into some kind of disease, it can be removed, and this has given this has given rise to a concept which is known as genetically engineered babies or three-parent babies that we are going to discuss now. All right. So the next concept we are going to discuss is nothing but a three-parent baby. So a three-parent baby is consisting of the mother, the father, and a donor mother. All right, I'll tell you that these three people are. Now consider the scenario that the egg of the mother is having 23 chromosomes in the nucleus. However, the mitochondrial DNA is having a gene which is possibly that may possibly result into emergence of a disease. So what will happen if this egg is fertilized by this sperm and the fertilized egg grows up into an individual? Then this individual, because of synthesis of protein from the mitochondrial DNA, will experience development of disease in later life. So now what the doctors have done is, doctors have devised a way in which the fertilized nucleus can be taken out of this unhealthy cell having diseased mitochondrial DNA, and it can be put into a donor egg. This donor egg is coming from the donor mother's egg. She is the third participant in this process, and therefore it is known as three-parent baby. So what you do is, the donor mother's egg is also containing the mitochondrial DNA and the nucleus. So what you do is you remove the nucleus from the donor mother's egg, and you leave the donor mother egg without nucleus but having mitochondria. And then what you do is you take the fertilized nucleus from the mother's egg, which was having diseased mitochondria, and transplant it into the healthy nuclearless donor mother's egg. So as I told you that the mitochondrial DNA do not participate in genetic material or genetic process, therefore there is no issue associated with genetic characteristic of the three parent baby. Rather, the baby will be having characteristics only from the mother and the father side, because the nucleus is from the fertilized nucleus is that of the mother's egg and the father's egg. Alright, and mitochondrial DNA is only the healthy thing that has been taken from donor mother's egg, and when this fertilized egg grows up, the individual will be all hale and hearty. Alright, so this concept is known as the three parent baby concept. Now we are going to discuss yet another important concept, and that is stem cells. Alright, so stem cells. Stem cells are those, so if you talk about the cells of the human body, the cells of the human body can be divided into two types. The first is the differentiated cells, and second is the undifferentiated cells. The differentiated cells are those cells which are specialized to form a particular type of tissue or organ. Alright, so like for example, your hair follicles or your skin cells. Your skin cells will keep on replicating into just another type of skin cells. They will not replicate into some other organ or some other tissue. These kind of cells are known as differentiated cells. However, there are also certain cells which have the potential to develop into different type of organs or tissue, and these type of undifferentiated cells are known as stem cells. So stem cells can be like, for example, there is a stem cell. That cell has the propensity or probability to develop into a liver cell. That cell has the propensity or ability to develop into a neuron. It can develop into a cardiac muscle. It can develop into n type of different cells, and therefore these cells are known as undifferentiated cells, and stem cells are the undifferentiated cells. If you talk about stem cells, there are two types of stem cells in human body according to their origin. The first is the embryonic stem cells. Embryonic stem cells are those stem cells which are present in the embryo. You know that embryo is nothing but a evolved fertilized egg which is fertilized by the sperm from the father, and now this embryo body will gradually develop. That is, the stem cells, some stem cells will develop into heart muscles or heart, some stem cells will develop into kidneys, some stem cells will develop into skin, some stem cells will develop into bones. That means it is the stem cells in embryo that are giving rise to the entire creature. So the cells present in embryo are known as embryonic stem cells. However, sometimes using some mechanism, stem cells are generally present also in human beings. They are known as somatic or adult stem cells, and sometimes your normal somatic cells, they can be through electric current provided a pluripotency, and that are known as IPSCs or induced pluripotent stem cells. Alright, so these are the two type of stem cells. Now let us cover what do you understand by the term cell potency. Cell potency is a very important term. What happened that sometimes there is a concept that the stem cells have the possibility to grow into differentiated and specialized type of cells. Now to how many number of different type of cells that can emerge from the stem cell is known as the potency of the stem cell. So stem cell in a way, um, cell potency is nothing but it is the varying ability of stem cells to differentiate into specialized cell types, and you can also arrange the cell types into the hierarchy of potency. For example, there are totipotent or omnipotent stem cells. The totipotent or omnipotent stem cells can give rise to any of the 220 cells that are found in the embryo as well as placenta. So these totipotent and omnipotent stem cells, they have the highest potency, that is, they can convert into any kind of cells that can be found in human body. Now then, there are pluripotent stem cells. These are those stem cells that can convert into any of the 220 cell types of the human body, but not the placental cells. Placenta is like, for example, in embryo, the embryo develops a feeding mechanism with the mother through a tube-like structure, and this is known as placenta. So placenta cells can be developed from totipotent cells but not pluripotent cells. But all other cell types can develop from pluripotent cells. And then there are multipotent cells. Multipotent cells can develop into different cell types, but they are fewer than 220. Alright. So if I arrange the decreasing order of cell potency, highest level pe aayega totipotent or omnipotent, then it will come as pluripotent, and then as multipotent stem cells. So that was all about the basics of this biotechnology. Now I'll conclude the lesson. That's all from the lesson. Now we'll be covering the important programs, policies and projects related to biotechnology in and around the world that is important from CSA 2020 point of view in our sixth lesson. So keep tuned for the sixth lesson. Be indoors, be safe, and prepare hard for your goal. That's all from my side. Have a great day ahead. Take care. Bye.